Welcome everyone to a brand new lecture series where I'm going to discuss about the molecules that make up our life. In fact, make up the entire us. That is me, you and everyone. Just pondering about it, it fills me up with a sense of wonder about how just mere molecules, their interaction, their communication, their lifetime partnership, I don't know how, but it makes the life possible that is bursting with colors of uniqueness and joy. With this deep sense of wonder, which I am sure is within you as well, let's start analyzing these magic molecules in our new lecture series entitled The Molecular World, or indeed the Molecular Biology. Now, starting with the magic molecules, let us start analyzing with the most magical molecule that makes us, that is the molecular map of who we are, that is each and every characteristics which accumulate together to make the person we are is actually mapped in a very tiny magical molecule known as a DNA. Now here in this series we are going to discuss and analyze the DNA replication that takes place in cellular organisms, and that too in those cellular organisms that do possess a double-stranded DNA. So without any other further delay, let's begin our session. So let's begin our journey by analyzing one of the very magical process in the molecular world, that is DNA replication. Here we are going to analyze the DNA replication that takes place in the cellular organism that do possess the double-stranded DNA. Now while doing so, we can categorize the replication process into two distinct categories, that is the DNA replication which takes place in eukaryotes and the DNA replication that takes place in prokaryotes, specifically in the eubacteria. Now talking about the DNA replication that takes place in the eukaryotes, before moving ahead we need to know one thing, that DNA or a genetic material is not only present in the nucleus, which is our basic knowledge, but also is present extranuclear, that is outside the nucleus, in certain cellular organelles, say for example in mitochondria, in chloroplast. And accordingly, the DNA which is present in the eukaryotes can be categorized as nuclear DNA and extranuclear DNA. In nuclear DNA we have our chromosomal DNA, which is linear. Now, how linear DNA construct the chromosomal DNA, the story we all know, where the linear DNA supercoil themselves to form a very special structure known as the chromosome. Hence, the structure which is formed by the linear DNA is known as the chromosomal DNA. Now, coming to the extranuclear DNA is mostly circular in nature. Now, having understood these eukaryotic characteristics of DNA, let's move on to the eubacteria. Now, in eubacteria, again the DNA can be categorized as chromosomal DNA and plasmid DNA. Now, chromosomal DNA in case of eubacteria are mostly circular. And talking about the plasmid DNA, now what basically are plasmids? Plasmids are small circular independent DNA which are capable of independent replication and is present mostly in bacteria. And to add an extra information, then I would love to say that it is very easy to manipulate the plasmid, and therefore the molecular biologists use plasmid in various welfare activities of the humans. And if you want to know more about the plasmid, please do comment. I will make sure that I make an elaborate video on plasmid. But for now, we need to understand that plasmid is a small circular DNA which is capable of independent replication. Okay, having understood a bit about the nature of the DNA which is present in eukaryotes and eubacteria, let's proceed further by digging deep into the replication process that takes place in the eubacteria. After discussing it, we will come to eukaryotes, but start with the small and then we will find the bigger picture as we move along. So, the DNA replication in eubacteria. Basically, we are starting with the chromosomal DNA. Now, before starting it, we need to choose a representative through which we can analyze the replication process in this group of organisms.
and therefore the representative which we have considered is a very famous representative and is very familiar to all of us, that is E. coli. And we all know that E. coli is a gram-negative bacteria. Talking about a few specifications of E. coli, then I would love to say that the number of chromosomal DNA which is present in E. coli is 1, and the chromosomal DNA which is present in E. coli is circular in nature. And talking about the size, it's about 4.6 into 10 to the power 6 base pairs, or in other words it's 4.6 million base pairs. And the mode of replication it chooses is semi-conservative mode of replication, where the newly formed DNA in the newly divided cell contains one strand of newly synthesized DNA and the other strand of the parental DNA, and that process of replication is known as a semi-conservative process. Now, having understood this as well, okay, just give me a minute. Let's understand how the semi-conservative mode of replication takes place. Now, in the semi-conservative mode of replication, the melting and replication do occur simultaneously. That do mean that it doesn't happen in this way, that first of all the DNA strand would be melted into two different strands and then the replication would begin. Now, it doesn't happen in that particular way. The thing is that both the melting and replication, that is both the strands would be melted and the replication will start simultaneously at the same time. Both the strands indeed replicate simultaneously. It is not that one of the strand will replicate first and then will be followed by the second. No, it doesn't happen in that way, but both the strands do love to replicate simultaneously. Replication occurs from 5' to 3' end. And the DNA polymerase. Okay, before moving to this particular statement, let us make sure that you understand this particular statement very clearly, that replication occurs from 5' to 3' end. Now, how the 5' and the 3' end is decided? For that, let's look at this diagram. Now here, this is a schematic diagram of a DNA. This is a linear DNA which I have drawn. Or in other words, I can say that this is a part of the circular DNA which we are discussing, a very small segment of the circular DNA, and this segment looks like this, right? Now here you can see these are the base pairs. These are the base pairs. Now these base pairs, they interact with each other via hydrogen bonding. Now look at this particular portion. If we zoom in into this particular segment, we can see that it looks somewhat like this. Don't be in a kind of panic by looking at the molecular structure. This is very easy. I will explain it to you. Now here it is the nitrogenous base, where I put A which resemble adenine. Here is the other nitrogenous base, right, which resemble thymine, and with this nitrogenous base a sugar molecule is attached, which is the deoxy form of sugar. Why it is deoxy form of sugar? Because in the second position, this is the first, this is the second, in the second position we have eliminated the oxygen, so therefore it is a deoxy form of the sugar. This is the third position of carbon in the sugar, this is the fourth, and this is the fifth carbon. Now observe very carefully. Now if the fifth carbon do hold the phosphate group, if the phosphate group is binded to the fifth carbon, that is this particular carbon, then this particular end would be considered as the 5' end. Now coming to this, now see, this is a phosphodiester linkage. This is a phosphodiester linkage, and here you can see this is the nitrogenous base, this is the deoxyribose sugar, this is the phosphate molecule. Now here if you see, then this is the first carbon, this is the second carbon, this is the third carbon, and here we can see that the third carbon is free, and wherever the third carbon is free, now see, a DNA segment may have two ends or may not, it must have two ends, where one end will have a free OH end, where the 3' OH is going to be free, and the other end, exactly the other end, will have the fifth carbon associated with the phosphate. So the particular end which holds the 3' OH that is known as the 3' end, and the particular end which holds the phosphate associated with the 5 carbon or the 5th carbon is known as the 5' end. Hopefully I think you have understood that what is the basis for categorization of 5' end and 3' end. Once again I would like to say, if there is an end where we can see that the 3' OH is free, then we can consider that particular end as 3' end. And if there is an end where we can see the 5th carbon of the sugar molecule is associated with a phosphate, then we can term it as the 5' end. And replication occurs from this end to this end, not the other way around. Now DNA polymerase, I think everyone know that what DNA polymerase is. It is the main protagonist enzyme which plays a very prominent role, a solid role in DNA replication process. But the sad thing to know is that DNA polymerase cannot initiate the process of replication, but it can only elongate the process of replication. Now if DNA polymerase cannot initiate the process of replication, who does? For this information, stay tuned for the upcoming videos. There we are going to reveal that who is the one who initiates the DNA replication process. So with that, let's wrap up our session. The next session is coming very soon. So if you have liked this video, please do subscribe, share, and wait for the upcoming session. It's going to be a very interesting session that I promise you. Thank you very much.